Hi everyone, I hope you've been enjoying Biodigital so far, and thank you for joining my presentation. My name is Haas-Kwon and I'm part of the marketing team here at Benchling. Today I'll be talking about how to accelerate innovation in the biotech and biopharma industries using modern R&D informatics. Before we get started, I'd like to share a quick snapshot of Benchling as a company. We were founded out of MIT back in 2012, and our first product was a molecular biology design suite. After many meaningful partnerships with leading companies in the life sciences space, we built out a total of 7 applications on a unified platform. Today, we're over 200 employees strong, support more than 300 enterprise customers ranging from rising startups to the top life sciences companies in the world, and have received funding from notable firms like Andreessen Horowitz, Thrive Capital, and Benchmark. All the while, we've remained true to our roots, supporting more than 215,000 academics worldwide. We were founded in the wake of a new, rapidly shifting era of medicine, where biopharmaceuticals have begun to dominate the landscape. The sheer pace of this innovation has been staggering. It's hard to think that gene editing as we know it today simply didn't exist just 10 years ago. Now, gene and cell therapies are delivering on the promise of personalized medicine, and biopharmaceuticals as a whole have grown from 15% to 50% of the biopharmaceutical R&D pipeline. In short, the future of medicine is no longer on the horizon, it is here and now. But despite breakthroughs in scientific technologies, there is still a lot of work to be done. Longer R&D cycles have led to faster technical obsolescence, as patents no longer provide the same safeguards as before. Molecules of interest are vastly more complex, and often, as with cell and gene therapies, the process is the product itself. And throughout all of these shifts, research groups are no longer producing gigabytes of data, but terabytes. All in all, the speed and complexity of biopharmaceutical R&D has been punishing. Of the thousands of medicines in preclinical development, we are just now starting to see the first cell and gene therapies reach the market, with Kymriah, the first CAR-T therapy approved in 2017, and Luxturna, the first gene therapy approved only last year. And this transition from small molecules to biopharmaceuticals hasn't been a clean break. Companies are blurring the lines between chemistry and biology with things like synthetic peptides, mRNA therapeutics, and ADCs. The game has changed, and pharmaceutical companies are tackling a new level of complexity in their processes and products. And with that territory has come vast, multiplex data sets. Organizations are beginning to embrace a new paradigm that leverages this data to inform their R&D strategies and shorten time to market. But how do these companies stay on top of all this information and use it to drive rather than hinder their businesses? In order to take advantage of this new R&D paradigm, companies need a new level of biointelligence to power their R&D and help manage the complexities of biopharmaceutical research. At Benchling, we think of this biointelligence as the interplay of sample intelligence and process intelligence. In large molecule R&D, this translates to end-to-end traceability of every sample all the way back to the sequence level, enabling smart lineage detection, and implementing automatic entity validation when creating or vetting candidates in their data. On the process side, this means full traceability of experimental conditions and results. Now, these are straightforward concepts. However, to date, they've proven to be the bane of nearly every R&D organization, as old and legacy systems simply cannot capture the level of granularity needed to produce this type of visibility. But imagine the challenges you could address if you had access to a context-rich database with all these components. As a scientist, you could quickly identify key results across your team or understand the history of a particular set of samples. As a manager, you could maintain a bird's-eye view over your team and hone in on process bottlenecks. Or even as an executive, you would have the power to analyze past and current objectives and build forecast reports using data produced directly at the bench. No matter where you stand within your organization, you need clean data to answer tough questions and inform your actions. But in most companies, there's no way to get this. So scientists end up repeating experiments and wasting resources. Managers often make decisions based on their best guess or on incomplete data. Or even in the best case scenario where all of the data has been properly recorded, it takes days or even weeks just to track it all down and turn it into something meaningful. Producing this level of biointelligence is a difficult technical problem. That's why we built Benchling from the ground up to tackle the hairy challenges at the forefront of biopharmaceutical R&D. As a unified biointelligent platform, we drive the creation of higher quality data that powers your biopharma programs. The Benchling Life Sciences R&D Cloud has 3 parts to it: an application layer, a data layer, and the underlying platform. Up top, you'll see a suite of applications for scientist productivity, sample tracking, and process management. In a nutshell, we combine the functionality that traditionally exists across separate ELNs, molecular design tools, LIMS, and project management software. Underpinning this is our data layer, built with native support for common life science data types like DNA and protein sequences, as well as the ability to define custom biological objects. In addition to the structure and standardization this provides, the data layer also models the relationships between entities with what we call biointelligent links. For example, a cell line can be linked to the plasmid it contains and subsequently to the protein it produces. Finally, our applications and data layers are built on top of a robust enterprise platform that allows for easy integration, customization, and extensibility. This is where Benchling marries native platform data with instruments and third-party software via REST APIs, and where the SQL warehouse connects to business intelligence and data visualization tools like Tableau and Numeracy. The net result is a single system for your scientists to use in their day-to-day work, and a single unified data model that allows you to centralize, standardize, and interconnect all of your R&D information into one place, from DNA sequence data to freezer inventories to experimental conditions and results. We've become a trusted partner to some of the leading biopharma companies, many of whom are actually here at BioDigital. They range from startups just getting their lab operations up and running, all the way to some of the world's largest pharmaceutical companies. The flexibility of our platform allows for a wide range of use cases, but as you can see here, we've done particularly well in a few well-studied areas such as gene editing, immunotherapy, synthetic biology, antibody engineering, and industrial and agricultural biotech. To shed some light on how we help these organizations, I'd like to walk through a high-level use case from one of our partners, Prebenchling. This workflow describes a typical fermentation run. Historically, an organization would have to plan experiments in their notebook, design their constructs in a molecular design tool, and leverage instrument control software to initiate a run. Then they would have to recover the resulting data in the form of spreadsheets, make sense of it, and then manually transfer it into their ELN and LIMS systems. Finally, they would run assays against these samples and store this information in various text files like Microsoft Word or Google Docs. This process was riddled with gaps and pockets for error, including several copy-paste steps across 4 different applications, manual data transfers and quality checks, and no sample ID verification. Now let's take a look at the same workflow with Benchling. Here, experiments and constructs are created in Benchling, and information is pushed into the controller software. After the bioreactor completes its run, that information is pushed directly back into Benchling and appended to the same samples and notes where they originated. Overall, this process provides much more efficient and accurate data collection by eliminating copy and paste and manual data transfers, mitigating the number of tools that the data touches, and automatically validating incoming data against a contextual database. Thanks to this unified approach, Benchling has become the most widely used solution for biologics and biopharma R&D. Our commitment to science, our dual approach to support scientists and their IT groups, and our ability to produce unprecedented insights and business intelligence have made us indispensable to our partners. Most importantly, our partnerships truly are partnerships. Unlike most vendors you probably worked with, we don't just sell you software and then walk away, but rather we work with you to actually deliver results. Our customers receive dedicated members of our support team, chock full of PhD scientists and published researchers, to closely understand your R&D environment and advise best practices for modeling your unique workflows and data. Finally, I want to leave you all with some of Benchling's recent endeavors. You can't dedicate yourself to advancing life sciences without committing yourself to the ebb and flow of modern health. In light of the recent COVID-19 challenges, we've launched a handful of initiatives to support those on the front lines of the epidemic. We've compiled a number of COVID-19 related resources, from sequences to protocols, to equip and empower scientists to take on their own research. We also realize that our network of Benchling users is vast, and we've generated channels across our academic and industry users to donate supplies to their local testing centers and healthcare facilities. Lastly, we recognize that one of the biggest challenges today is the accuracy and availability of testing. We're hoping to alleviate some of the difficulties associated with this process through our platform. If your lab is working on COVID-19 diagnostic testing, we're giving away our full platform for free. And with that, I'd like to conclude by thanking you once again for listening in. Please don't hesitate to reach out and set up some time if you're interested in learning more about us and our partners. Take care.